To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.
Right, children. Now we are going to start the second chapter of this lesson. So in the first chapter, we learn about convex lenses and hand lens or the simple microscope. Now we are going to learn about the compound microscope, compound light microscope. Okay? So what is the meaning of a compound light microscope, children? Now when we consider about the simple microscope contains only one convex lens. It's known as the hand lens or magnifying glass as well. Right? We'll go through this. The compound light microscope is made of at least two convex lenses. Now, do you remember in the last activity, we used two hand lenses to observe or distinguish two dots. So there you observe very clearly when we use two hand lenses, we can get a better magnification and a better resolving power. Right? So using that method, compound light microscope is made. Okay? The compound light microscope is made of at least two convex lenses. Right? Two convex lenses. So why do we call it compound microscope? Because it has two convex lenses. Simple microscope or the hand lens has only one convex lens. This microscope has two convex lenses. That is why it is known as compound, right? Therefore, it is called the compound microscope. Why do we call it light microscope? Because in order to observe a specimen or an object, we need to get the help of a light beam. Okay, children, that is why it is known as light microscope. So we can put those two words together and it becomes compound light microscope. Compound microscope because it has two convex lenses. Light microscope because it needs a beam of light to observe the specimen. Okay? So light microscope now. Therefore, it is called the compound microscope and it is also called the light microscope because the light is used. Okay? Although microorganisms cannot be observed through a hand lens. Now we observe different samples using hand lens. Could we observe microorganisms? No. Because microorganisms are too small to be observed using a hand lens or a simple microscope. Now we cannot observe microorganisms through a simple microscope, but using a compound microscope, we can observe microorganisms as well. Right? So using the simple microscope, we can magnify or we can get a bigger image of a smaller objects. Right? But when you use the compound light microscope, we can magnify the objects or the things that cannot be seen by the naked eye. Right? So although microorganisms cannot be observed through a hand lens, they can be observed through a compound light microscope. What are the different types of microorganisms that you know, children? There are bacteria, there are fungus, unicellular organisms, so protozoans, viruses are there, right? Out of all these microorganisms, remember, we can observe the microorganisms under the compound light microscope, but viruses cannot be observed. Right, children? Because viruses, when we compare the viruses with the other microorganisms, viruses are way too small. So even the compound light microscope is also not powerful enough to observe the viruses. Right? So at the end of the lesson, we are going to learn about the electron microscope. Using the electron microscope, it's more powerful than that of this compound light microscope. So using the electron microscope, we can observe the viruses. So here, all the other microorganisms apart from viruses, we can observe under the compound light microscope. Right? Although microorganisms cannot be observed through hand lens, they can be observed through a compound light microscope. So needless to say, if we can observe microorganisms through compound light microscope, we can come to a conclusion that magnification and the resolution are way higher than that of the naked eye and the hand lens. Okay, The maximum magnification of a developed compound light microscope is about 2000 times, right? It's about 2000 times. The maximum resolution is about 0.2 micrometers. How do you write micrometers? Micro. This letter is known as mu, right? Micrometers, right? What is it in millimeters? 0.0002 millimeters. What is the resolving power of the naked eye? That is 0.1 millimeters, right? Here resolving power is way higher because it is 0.0002 millimeters, so 0.2 micrometers, right children? So maximum magnification of a developed compound. There are different types of compound light microscopes, right? The developed compound light microscope's magnification is about 2000 times, right? The resolution or resolving power is 0.2 micrometers or 0.0002 millimeters. Right, children? That is why we can observe microorganisms under the compound light microscope. Okay? Right. So we have this activity to do. Let us identify the parts of a compound light microscope. So when we learn about the compound light microscope, this is an expensive object, expensive instrument. So you have to handle this instrument with care, right? So in the lab, your teachers also will show you what are the parts of a compound light microscope, okay? So observe the compound light microscope in your school laboratory. Identify its main parts and functions with the assistance of your teacher. Now we are going to go to the lab to observe the parts of a compound light microscope. Right, children. Now I'm going to show you all the parts of a compound light microscope. Now look at this. This is the compound light microscope. But remember, this is a developed compound light microscope. This is a modern one. Most of our schools have the basic type of compound light microscope. It is a bit different than this, right? So I will show you what are the differences and all, but basically all the parts are there, which is similar to the basic compound light microscope, okay? Right. Now you all know that we call this unit a compound light microscope. Why compound? Because we look through two lenses, right? When we look at the specimen or an object, we look through two lenses. Where are those two lenses? This is the first lens. Now here you all can see there are two lenses, two sides here, right? To keep both the eyes we have. Now normally in basic compound light microscope, only one tube is there, tube-like structure is there, right? You all can see here. Here there are two parts to keep both the eyes, right? So this is a modern microscope. So using both the eyes, we can get a clear and a image with 3D effect. That's the importance of using these two. But most of our schools have the microscope with only one of these type of tubes. Now this is the eyepiece. This is the first lens. This is the lens that is closer to the eye. We can remove it like this. Okay, this is the eyepiece. Right, children? Right. Where's the second lens, children? Second lens is known as the objective. This is the objective, children. Now here, objective lenses are there in three different sizes, magnifications, right? Now this one, as this is a modern compound light microscope, this has 4 sizes. You all can see. Now I can rotate it. You all can see very clearly. I can rotate it. So whenever we need, according to the need, we can rotate and we can change it to the required objective. Now why do we call this objective? Because this lens is the closest to the object or the specimen. Now this is where we place the specimen, right? This is where we place the eyes, keep the eyes. Now this lens is the closest to the eye, therefore this lens is known as the eyepiece or eyepiece lens. right? And these lenses are closer to the object, therefore they are known as the objectives. And remember, we can change the objective as we need, like this. The lowest power, now here we have 4 lenses, but normally in basic microscope we have 3 lenses: low power, medium power and high power, right? Now these lenses are connected to a plate-like structure. This can be rotated. This is known as the nose piece, right children? So in a basic microscope, this part is known as the body tube. This whole part is known as the body tube. The shape is a little bit different, right? But this is the similar thing. So now, what is this part, children? This is called the stage. This is where we place the specimen or our object. Now here I have a slide. This is a permanent slide with a specimen. Now then I have to place this slide on this stage, and after placing it I have to fix it with this stage clip. It will not move here and there then, right? In a basic microscope there are 2 stage clips on either side, but here, because this is a modern one, it's a bit different, right? You all can see here. Now it will not move here and there. It is nicely fixed on the stage. Right, children? Okay. Now, can you all see under the stage, there's this circular part, children? This is known as the diaphragm. Now, you all know we call this compound light microscope. What is the reason? Because it uses, why light microscope? Because it uses a beam of light in order to observe the object, right? So light is coming from here, but at times we need to control the amount of light that comes towards the specimen. So that is done by this diaphragm. Okay, children, by rotating this diaphragm, we can adjust the amount of light that comes. Right. Now look at these 2 knobs, children. You all can see, you all can rotate it. Now this bigger knob is known as the coarse adjustment knob. Now what happens when I rotate this one? You all can see in this microscope, the stage moves up and down. But in the basic microscope in the lab, what happens, children? When we rotate this one, this body tube moves up and down. Okay? Either way, what happens, children? The distance between the stage and the objective lens can be increased and decreased by rotating this big knob. Right? By rotating the coarse adjustment knob, we can change the distance between the objective lenses and the stage. Here in this modern microscope, when we rotate this coarse adjustment knob, what happens, children? This stage moves up and down. But in the basic microscope, what happens? This body tube moves up and down. Right, children? What is this small one? You can't see. Even though when I rotate this one, you can't see any change. Even though you can't see any change here, this distance is finely changing. Right? So for the final or the fine changing of the distance between the stage and the objective lens, we can use this smaller knob. This is known as fine adjustment knob. Right? Coarse adjustment knob, fine adjustment knob. Right, children? Okay. So what is this one? This is known as the arm of the microscope. So when we carry the microscope, when we move the microscope from one place to another place, this is very important. Not only that, this is known as the base. Right? This is the base. We have to make sure when we carry this one hand should go under the base, and from the other hand we have to hold this arm. Right, children? Now look at this one. What is this, children? Now this is a developed microscope, as I told you all before, right? And at the same time, you all know that this microscope needs a beam of light in order to observe the specimen. Here, this is a light bulb. Now there's a wire connected to this microscope and there's a switch here, right? So when I turn on the switch, you all can see there's a light. Okay, children, I will switch it off. can see it goes off. Now again, I turn it on. Right, children? Now in the basic microscope, remember this place, instead of the light bulb, what is present, children? There's a mirror. That mirror can be rotated. One side of the mirror is a plane mirror, the other side of the mirror is a concave mirror. Right, children? That mirror can be rotated. What is the importance of that mirror, children? When now this developed microscope, this has a light source fixed to the microscope itself. But in that basic microscope, there's no light source. Therefore, light coming from the other side should be directed towards the specimen. That is done by the mirror. You all know that mirrors can reflect light. So we can, by adjusting the mirror, by rotating the mirror, we can direct light coming from one side towards the specimen. So that is the importance of the presence of the mirror in the basic microscope. But does this microscope need a mirror? No, because this comes with a source of light. Okay, children, this comes with a light bulb, therefore it doesn't need another mirror to direct light onto the specimen. So I hope you understood about the microscope, and now you know all the parts of a compound light microscope. Right, children? So you observed the parts of the compound light microscope. Now you should be able to label the parts by yourselves. Let's do this now. Right. Parts of a compound light microscope. So what are the parts, children? Now before I explain this one, label this by yourselves and then watch this one and check whether all the parts you labeled were correct or not. Now I already explained this in the lab. What are the each parts and even their functions. Okay. Let's try to do this. Right. So if you have completed this one, now you all can help me to label this. Right. We'll start from here. What is this, children? Now we know that in a compound light microscope, two convex lenses are used at once. Right, at a time, two convex lenses are used. One of these convex lenses is placed here. So this is where we keep our eye on. This is known as the eyepiece, right? Eyepiece. Eyepiece. Right? What is this, children? I showed you all in the lab. In order to move this part up and down, we can use this. There are two knobs. One is bigger than the other, right? This is known as the coarse adjustment knob. This is known as the fine adjustment knob. So when you use this one, we can clearly see this part goes up and down, right? In some microscopes, this stage goes up and down, right? In some microscopes, when you move this one, this part is known as the body tube. We label that as well. Body tube, right? When you rotate this coarse adjustment knob, in some microscopes, this body tube goes up and down. In some other microscopes, this stage goes up and down, right? Okay. We'll first draw the arrow as well. We label this as well. Even though it's not labeled here, this is known as the nose piece. Right. So this is the coarse adjustment knob. And this is the fine adjustment knob. Fine adjustment knob. What is this, children? This is the arm. Arm is used to hold the microscope, carry the microscope from one place to another place, right? This is called the arm. This is the stage. This is where you place the specimen on, and using the stage clips, you can fix the specimen firmly onto the stage, right? Stage clips. What about this? This is the base. This is the body tube. This rotatable plate is known as the nose piece. So the other lenses, now there are two types of lenses. One lens is the lens that is closer to the eye. The other lenses are closer to the object or the specimen. Those are known as objective lenses. There are mainly three types of objective lenses: mid power, high power and low power. Right, children? So these objectives are fixed to a rotatable plate. That plate is known as the nose piece. Nose piece. And these are objective lenses or objectives, also correct. Objectives. What is this, children? Right under the stage, there's a part called diaphragm. Now, diaphragm is used to control the amount of light that is coming towards the specimen, right? Diaphragm. And this is the mirror. Do you remember in the 9th lesson we learned that in the light microscope, mirror is used. There are two types of mirrors. One side plane mirror, the other side concave mirror, right? So this mirror can be rotated. This is the mirror, right? So parts of a compound light microscope: eyepiece, coarse adjustment knob, fine adjustment knob, arm, stage clips, stage, base, body tube, nose piece, objective lenses, diaphragm, and the mirror, right? Now we are going to see what are the functions of each part, right? Now here we have to write the part. Even this one you can try before me, okay? This side you have to write the part of the microscope, and this side description, the functions, right? So the closest lens to the eye, it is a convex lens. Magnification is shown as, it depends, right? From microscope to another microscope, it depends, right? Shown as 5 times, 10 times. How do you write the number of times? You have to write the multiplication mark and the number, right? So when you say this, it's shown as 5 times, 10 times or 15 times. You write the number with the multiplication mark, right? The specimen is magnified by using them, right? So here what is this? This is the eyepiece, because this is where you keep the eye at this place. Therefore, eyepiece is the lens that is closest to the eye, right? So let's write eyepiece. Next one, the closest lens to the specimen. What are they? Objectives, right? They are made of convex lenses. It magnifies the specimen, right? So the specimen gets magnified 2 times. First through the objective, then through the eyepiece, right? Mostly there are three objective lenses that can be identified. They are low power objective lens. That is the lowest power. Low power objective lens, the lens with the least magnification. It is about either 4 times or 5 times or 8 times, right? And then mid power objective lens, medium power or mid power. The lens with the medium magnification, mostly 10 times, right? And high power objective lens, right? The highest power, the lens with the highest magnification, it's about 40 times, right, children? So whenever you observe any specimen under the compound light microscope, you can choose which lens that you are going to use according to the specimen. If it's too small, you have to use high power. Otherwise, some of the specimen, even the low power is also enough, right? So mid power lens, low power lens and high power objective lens. So what should we write here? Objectives or objective lenses. Objective lenses. Right, children? Okay, next ones. To adjust the body tube to get a clear image of the specimen. Basically, to adjust the body tube, to move it up and down, we use the cause adjustment knob. To adjust the body tube finally, now when you use the fine adjustment knob, the smaller knob, you can't see the body tube moving, but it's finally just gets adjusted, right? So to adjust the body tube finally to get a more clear image of the specimen, right? So this is the cause adjustment knob. Cause adjustment knob. This is the fine adjustment knob. Right? Next one. To keep the slide of the specimen, where do you place the slide of the specimen on the stage? And to keep the slide of the specimen on the stage, fixed at one place, you use the stage clips. Stage clips. To control the amount of light obtained by the specimen, right? So the light coming from the under, you can control using the diaphragm. Diaphragm contains concave and flat surfaces. Focus light onto the diaphragm. Now I told you when it comes to the mirror of the microscope, it has two sides, right? You can rotate it over this place. You can see from either side it's fixed like this, it's circular, you can rotate it, right? One side is flat, which means a plane mirror, the other side is concave mirror, okay? So it is very important to direct light. Now you all know that in order to observe specimen under this microscope, we need to get the help of a light beam. So light is directed towards the specimen, towards the lenses using that mirrors. That is because mirrors can reflect light, okay? So contains concave and flat surfaces, focus light onto the diaphragm, that is the mirror. And finally, keeps the microscope on the supporting surface steadily, that is the base. Right, children? We'll go through this again. Eyepiece, the closest lens to the eye. It's a convex lens. Magnification is shown as 5 times, 10 times or 15 times. The specimen is magnified by using them, right? Objective lenses, the closest lens to the specimen. They are made of convex lenses. It magnifies the specimen. Mostly there are 3 objective lenses that can be identified. They are low power, mid power and high power, right? Cause adjustment knob, to adjust the body tube to get a clear image of the specimen. Fine adjustment knob, to adjust the body tube finally to get a more clear image of the specimen, right? And the stage. To keep the slide of the specimen, we use the stage. And stage clips to keep the slide of the specimen on the stage without moving here and there. Diaphragm controls the amount of light obtained by the specimen. And mirror contains concave and flat surfaces, focuses light onto the diaphragm, right? And the base keeps the microscope on the supporting surface steadily, right? So now you should be able to label each part and you should know the function of each part of the microscope too. We'll move on to the next. Okay, so we have this extra knowledge part, okay, extra knowledge on these microscope, right? So a bit from the history. The compound light microscope was first made by Zacharias Janssen. This is the person, Zacharias Janssen, and his microscope. So compound light microscope means the microscope that uses two convex lenses. So this is the person who made this microscope with two convex lenses for the first time. But what he made was not a very developed one. The Zacharias Janssen was a Dutch spectacle maker, right? So the magnification power of it was only 9 times. At the same time, even though it's the magnification is very less, very low, still he introduced this one, and because of that the other scientists could develop that one and observe, or they could increase the magnification of the microscope. Okay, so here look at this one. But the honor of making a microscope first goes to the Dutch scientist Antony van Leeuwenhoek, right? Antony van Leeuwenhoek was the he's a Dutch scientist and he is known as the father of microbiology, right? Okay, so the reason is that a world not seen from a naked eye was revealed by this instrument. This was a developed type of microscope. Now when you look at the magnification power of this microscope made by Zacharias Janssen, it was only 9 times. So even though this person introduced the microscope, he made it for the first time, because of the low magnification power, it was not possible to observe the microorganisms using this microscope. But the microscope that was developed by Antony van Leeuwenhoek, it was possible to observe the microorganisms as well, which means he was the first person to observe the microorganisms under microscope, right? The reason is that the world not seen from a naked eye was revealed by this instrument, which means the microorganisms, right? You all know that using hand lenses, we cannot observe the microorganisms. Only using a compound light microscope, we can observe them. Right, children? So this is the person, Antony van Leeuwenhoek, and his microscope. Right, children? So we have an assignment to do related to this. Prepare a report on the history of the microscope. So here in this note, only about 2 scientists, 2 people who made microscopes are given. So you can read more articles, you can get the help of the internet, and you can, there are many articles on this topic, right? You can read more articles, and like there are other people who tried to develop this microscope, which was invented by Zacharias Janssen, right? So you can find more details about those scientists as well. Right, children? Or you can read books. Go to your school library and you can refer to books and you can find more information as well. Right? Okay, we'll move on to the next. The correct method to use a compound light microscope. So when we use this compound light microscope, as I told you all before, it is an expensive instrument. Therefore, we have to take care. We have to handle it with care. Right, children? So there's a certain way that you use it. So we are going to learn what are the ways that you have to consider, what is the correct method of using it. And after this, we are going to learn about the safety of the microscope, what are the steps that can be taken also. Right? So here, this is about how to use it, this instrument, how to use it properly in order to observe specimen, right? Number 1, First, keep the microscope on a horizontal table steadily. You have to find a flat surface to place this microscope on, right? In a place where strong sunlight will not fall. We need a beam of light in order to observe the specimen, but at the same time, if the sunlight is too strong, it is very disturbing, right? And at the same time, it might even damage our eyes too. Therefore, we have to place this microscope in a place where strong sunlight will not fall. Okay? Then, use the coarse adjustment knob to bring the low power objective lens down. Now, first of all, before we observe the specimen, first of all, we have to get enough amount of light towards the microscope. If only light is present, we can observe our specimen properly. Right, children? Therefore, first of all, use the coarse adjustment knob to bring the low power objective lens. When it comes to objective lenses, there are 3 powers present: low power, mid power, high power. Right? So, using the coarse adjustment knob, you have to move these objective lenses towards the stage. Okay? Adjust the mirror and the diaphragm to obtain a clear spot of light by keeping the eye open on the eyepiece. Right? Eyepiece is the convex lens that is closest to the eye. Okay? Remember, children, when you keep the specimen on the stage and the time you remove the specimen from the stage, both the instances, you should do it under the low power, which means we'll go to see this microscope. Right. Now, the low power is the shortest lens. Now, there are 3 lenses. Basically, there are 3 lenses fixed to this nose piece. Those are known as objectives, right? So, they are low power, mid power and high power. So, high power is the longest one, then the mid power, and the shortest one is the low power. Remember, when you place the specimen on the stage and when you remove the specimen from the stage, both the instances, you have to rotate this nose piece to low power. That is the shortest objective lens, right? So therefore, it will not get harmed because of the slide or the stage. If you are trying to remove this specimen, or if you are going to insert the specimen when the high power or the medium power is on under this one, right? Then what will happen? Sometimes it might, when you are trying to insert this slide with the specimen, it might damage this because they are longer than the low power, right? It might damage the lens. So what is the most important parts of the microscope? It's lenses. If not for the lenses, we cannot observe the microorganisms or any other specimen. Right, children? Therefore, we have to take a special care of these lenses, the convex lenses. Right? That is why whenever you insert the specimen or whenever you remove the specimen, don't forget to rotate this nose piece under low power to take the low power aligned with the eyepiece. Right? Because it's the shortest one, it will not harm the lens. Otherwise, because these two are way longer than the low power, when you are trying to remove these slides, glass slides, it might damage the lens. Okay, children? That is very important. Right, we'll move back. Right. So then fix the prepared slide on the stage using clips. Carefully place the slide on the stage and you have to fix it with the stage clips. Okay? Bring the low power objective lens upwards using the rough adjustment until a clear image is obtained. You have to do it slowly, right? Make sure that both eyes are open during the process and keep the comfortable eye at a distance of 1 centimeter from the eyepiece. Most of the time when we look through the microscope, unintentionally we close one eye, right? But this is wrong, right? Because in the upper grade, sometimes when you do science or biological science for your A-level, sometimes you have to draw the diagram of the specimen while looking from the microscope, right? So both the eyes should be open. So while looking from one eye, at the same time you have to draw the diagram. At the same time you have to do this. Therefore, closing one eye is not correct. Okay, children? So make sure that both eyes are open during the process and keep the comfortable eye at a distance of 1 centimeter from the eyepiece. Okay? Right. Then try to obtain a clear image using fine adjustment. Fine adjustment means the smaller knob. Right? A sharp and clear image could be obtained by adjusting the mid and high power objective lens if necessary. Even earlier I told you all, for certain specimens, using the low power is just enough. But sometimes we need to use the high power and mid power as well. If the low power is not enough, then we can go to the mid power. If the mid power is also not enough, then we can go to the high power. Anyway, don't forget to rotate it back to the low power when you are going to remove the slide from the stage. Right? Use the rough adjustment to bring the low power objective lens upward after the observation. Right? What you have to do after observing the specimen, you have to take the objective lenses up by rotating the rough adjustment. Okay? And finally, remove the specimen from the stage. Use a dry piece of cotton cloth to wipe the stage. Right? So after using, you have to make it dry. If the moisture is present, what will happen? Fungus will form on the lenses. So if the fungus forms on lenses, what happens, children? We can't use those lenses anymore. Therefore, you have to keep this instrument dry. That's why you have to wipe it with a clean or dry piece of cotton. Okay, children, we are going to the lab in order to take all these steps in the lab now. Right, children. Now I'm going to show you all what is the correct method of using a compound light microscope. Right? Now, first of all, we have to place the microscope on a flat surface. We have to make sure it is not tilted and the surface should be flat. Right? What are the reasons for this, children? Then it will not harm the microscope. Now, it might even fall off if we place it on a slanted or an angled surface. Therefore, when it is very steady when we place it on a flat surface. And another reason is, let's say that if you are observing a liquid specimen, in that case, when we use a slanted surface, what will happen? That liquid will spill off. Therefore, in order to avoid that one, we have to use a flat surface. Okay. Then what we have to do is, we have to bring the low power down. Low power. Now here it's already low power. Low power objective is down. So in order to move this low power up and down, we need to increase and decrease the distance between the stage and the objective. What do we have to use? We can use this coarse adjustment knob. Right? Okay. So we can move it like this if you want to take it towards the specimen, and otherwise to the other direction. Okay? Right. Then what we have to do? We need to adjust the amount of light coming towards the specimen. Now in a basic microscope, this is done by rotating the mirror, right? Turning the mirror to the specific direction where the light is coming from, and also using the diaphragm. Now in this developed microscope, do we need a mirror? No, because we are provided with a light source. Now here you all can see the light bulb, right? So we have a light source, therefore we can skip that step here. And using the diaphragm, we can adjust the amount of light that is coming towards the specimen. Right, children? Now what we need to do is, we have to look through the eyepiece. Remember, when we look through the eyepiece, we have to make sure that both eyes are opened. Okay? Right. And you should not take your eyes too close to the lens. Eyes should be at least 1 centimeter away from the lens. Okay? Now we need to place this slide. Now I have a permanent slide here. We need to place this slide. This is our specimen, what we are going to observe, right? We need to place this slide on the stage. Make sure whenever we are going to place this slide on the stage, always this low power should be aligned with the stage, right? Okay, children. This is how we do. And using the stage clips, you have to fix it. I already showed you all how to do that earlier. Right, children? So what is the reason we always place this low power line with this stage, children? Whenever we place the slide, and also when we are removing the slide, this should be done. What is the reason, children? Otherwise, now you all can see these are too long. These are longer. The other lenses are longer than this low power. So by the time we are placing this slide on the stage, if these are aligned with this one, sometimes when we are placing the slide, we might accidentally damage the lens. We have to protect the lens of the microscope. That is why always the shortest one should be aligned with the stage. Right, children? Now we need to observe our specimen using the microscope. So in order to get a clear image, we can use the fine adjustment knob, right? And sometimes if the image is, now if you want to get a the larger view, better view of the image in a certain particular part, then we can change the objective lens, right? Then we can change the objective lens by rotating this nose piece, right? So we have to be very careful when we do this one, not to damage the lens, right? And then while looking at this one, with the fine adjustment knob, we can get a clear image of a particular part of the slide. Right, children? So after observing, what do we have to do, children? Now we need to remove this slide, okay? Now what is the next step? Are we going to remove it like this? No, because this is not the lowest power. As I told you all, for the protection of the microscope, we need to put it back to the low power. Right? See, it is very short, and because of that, when we take this slide away from the slide, we will not harm the lens, okay? So I am going to take it out. And finally, after using this microscope, we need to wipe the stage with a clean piece of cloth or clean piece of cotton to remove any moisture. Right? What is the reason? Otherwise, if there's any moisture left, it will harm the lenses by forming fungus. Right, children? So we have to make sure that we nicely wipe off the stage with a clean piece of cloth or clean piece of cotton. So I hope you got a better idea about using the microscope correctly. Right, children? So you observed how to take all these steps. Now moving on to the next. Right. Factors to be considered in the use of a microscope. I told you all this is an expensive instrument, therefore we have to handle it with care. So for the safety of the instrument, we have to follow these steps. Let's see. When the microscope is carried from one place to another, the arm of the microscope should be held by your familiar hand. Look at this picture. This person is holding from its arm, right? With the familiar hand. Now this is a heavy instrument, okay? So with your familiar hand, and the other hand should be kept below the base of the microscope, right? Not with one hand. You have to use both the hands to carry it from one place to another place, right? One arm, you're comfortable, or your familiar hand, you have to hold this arm with that hand, and the other one should be under the base, okay? Then hold the microscope towards your body. You have to tilt it towards your body, so it's safer. When you move from one place to another place, it will not move here and there if you hold it towards the body, right? Number 2. Wipe the stage using a clean piece of cotton after using the microscope. Even earlier I explained this one, right? If moisture is present in the microscope, it might form fungus on the surfaces, especially on the lenses. Therefore, to prevent that, so you have to wipe the stage after using with a clean piece of cotton, right? If the microscope is not used for a long period of time, the lenses should be removed and placed inside a desiccator with silica gel or anhydrous calcium chloride. Why do we have to do this? What is a desiccator? A desiccator is a kind of glass jar. You can seal it. It is well sealed, right? It is a kind of airtight jar-like object, right? So it is well sealed. Okay? So what is the meaning of silica gel? What do they do? Silica gel or anhydrous calcium chloride? Silica gel and anhydrous calcium chloride, they can absorb. They are chemical substances. They both can absorb moisture present in the atmosphere. Right, children? Have you seen sometimes when you buy leather shoes or leather bags, a small silica gel packet is present inside? That is in order to absorb the moisture, and it will not, because if moisture is present, fungus will form on leather. To prevent this, that is why when you store your leather shoes or bags without using for a long time, it's better to store it in boxes or bags with silica gel on, because then even though moisture is present, it will be absorbed by silica gel. Right? In the same way, if you are not going to use this microscope for a long time, it's better to remove the lenses. Now, lenses can be removed, right? I will show you how to remove the lenses. So, the lenses should be removed and you have to place them in a desiccator. Now, desiccator is a kind of, as I told you all, it's a kind of airtight jar. So, you have to put silica gel or anhydrous calcium chloride inside this desiccator with the lenses, right? So even though there's moisture present inside the desiccator, it will be absorbed by this silica gel or anhydrous calcium chloride. Right, children? This is for the protection of the lenses. I told you all, lenses are the most important parts of the microscope. Okay? The microscope should be stored in an upward position, right? It comes with a special box. It's better to store as it is in the box in a vertical position. Upward position to minimize the accumulation of dust, right? Use clear slides and cover slips in the observation. In order to mount the specimen, in order to place the specimen, we have to use glass slides, and also to cover the specimen, we use cover slips, right? So the cover slips and glass slides should be very clean, okay? And the last one, do not change the lenses of one microscope to another. We should never ever do this, right? Let's say that if your school laboratory has more than one microscope, if there are several microscopes, don't ever try to change the lenses from one microscope to the other. I told you all, you can remove the lenses, but just because of that, you should not change it from one microscope to the other microscope. Why? In case if a certain lens has microorganisms formed, or especially fungus form on the lens, right? If fungus is present on a certain lens, when you change it to the other microscope, what happens? It will spread to the other microscope as well, right? So all these steps should be taken for the protection of the microscope, because it is an expensive instrument. We are going to lab to follow these steps now. Right, children? Now we are going to discuss the factors to be considered when using a microscope. Now, first of all, remember, microscope is an expensive instrument. Right, children? Therefore, we have to be careful when we handle the microscope. Right? So, what are the steps that we can take in order to protect this instrument, children? Number one. Now, when we carry the microscope from one place to another place, we should make sure both of the hands are used, okay? Now, the familiar hand should be always used in order to hold this handle. And then we have to keep the other hand under the base, like this, right? When we take it from one place to another place, then slowly you have to place it like this, and you have to keep it closer to your body and towards your body, right? So we can make sure, by doing this, we can make sure this will not fall off. We are not going to drop this even accidentally, right? So this is how we are going to carry it, with both hands, and the familiar hands on the handle, the other hand under the base, and then you have to hold it towards your body like this. And then when you go to another place, and you have to place it on another flat surface very carefully. Right, children? That is the first thing. And even earlier I mentioned this one. After using the microscope, we always have to wipe the stage with a clean piece of tissue or a cloth or even a clean piece of cotton. Right, children? And the next thing is, children, you all know that we are going to observe different types of specimen under the microscope. Okay, children? So sometimes we need to make our own specimen. So when we make our own specimen, we need to use slides and cover slips. Okay? So I'll show you. This is a glass slide. Right? So when we make a specimen, we have to place the specimen on the middle of the slide, and then we need to cover the specimen with a cover slip, this thin piece of transparent cover slip. Right, children? So remember, whenever we use these type of slides and cover slips, we have to make sure they are all clean. Right, children? We need to make sure they are all clean. Not only that, when we place this slide prepared by us on the stage, we have to first of all wipe it off. Right? If there's any water or other liquids, we need to wipe off the excess liquid of the slide. Okay, children? That is the other thing. And the other thing is, children, we should never change the lenses of one microscope to another microscope. Even earlier I showed you all, we can remove these lenses, right? So we should never ever change one of these lenses to another different microscope. What is the reason? Now, most of the time, these lenses get damaged by forming fungus on that one. So let's say that if this microscope, if this lens has fungus, when we change it to another one, the other parts of the microscope, that another microscope also will get fungus. Right, children? So fungus are microorganisms, they spread everywhere, right? That is why we should never change the lenses of one microscope to another microscope. Right, children? And the other thing, now, what is the most important parts of the microscope? The lenses, right? And lenses always get affected with fungus. Now, because the environment, there is always moisture, there's water vapor, right? When a lot of moisture is present, those places fungus can grow very well, right? So if we are not going to use the microscope for a long period of time, what do we have to do? We need to remove these lenses. We need to remove, all these lenses can be removed. So we need to remove these lenses and we need to store them in a special type of glass jar. It's an airtight glass jar and it is known as a desiccator. We need to remove all the lenses and we need to store them in a desiccator. Desiccator is it's a type of airtight glass jar. So we have to make sure inside this glass jar called the desiccator, either silica gel or calcium chloride should be present. Right, children? What is the importance of the presence of silica gel or calcium chloride, children? Because silica gel and calcium chloride can absorb moisture or water vapor present in the atmosphere. Therefore, in order to absorb all the moisture present in the inside the desiccator also, we have to put some silica gel or calcium chloride along with these lenses. Okay, children? So the next thing is, when we store this microscope in cupboard or anywhere, we always have to place this in an upright position so that it will not collect much dust. This type of modern microscopes come in a special case, so we need to make sure that after using this one, if you are not using for quite some time, we need to place it in that special box that it comes on. Right, children? So I hope you understood how to take care of the microscope in the lab. Right, children? So I hope that part was clear. Now you know how to follow these steps according to protect the microscope, right? Okay. Next topic is calculation of the magnification power of a microscope. Now, what is the magnification, children? Magnification means how many times a certain object gets magnified by a microscope or even a hand lens. Okay. So, how to calculate the magnification power of a microscope, children? Let's see. The magnification power means the number of times that the specimen is subjected to be magnified. Okay. The number of times is obtained by multiplying multiplying the magnification of the eyepiece with the magnification of the objective lens. Right? In a compound light microscope, there are two lenses. One is closer to the eye, the other one is closer to the object or the specimen, which means the specimen gets magnified 2 times, first by the objective, then by the eyepiece. Right, children? Therefore, when you calculate the total magnification of a compound light microscope, you have to multiply the magnification of the eyepiece by the magnification of objective lens. Therefore, magnification of the microscope equals, right, magnification of eyepiece into magnification of objective lens. Right, children? It's very easy to memorize. We have a calculation to do here, right? The magnification of eyepiece was 10 times. How do you write that? With the multiplication mark and the number. And the magnification of the objective lens was 40 times in a particular microscopic observation. What is the magnification of the microscope? Now calculate this one before me, children. I am sure, because now I explained you all how to calculate using this formula. It's very easy to do now, right? Okay, let's do this. First, we will write the formula. Magnification of the microscope equals magnification of the eyepiece into the magnification of the objectives. Right? Magnification of the microscope equals the magnification of the eyepiece into the magnification of the objective, right? So here it's given the magnification of eyepiece was 10 times. We'll do this 10 times into magnification of the objective lens was 40 times, right? 40. So what is the answer? 10 into 40, 400. 400 times. Magnification is 400 times, right? Magnification of the microscope equals the magnification of the eyepiece into the magnification of the objective. Magnification of the eyepiece is 10, objective lens was 40, right? 10 into 40, 400 times. The magnification of this particular microscopic view is 400 times. Right, children? Okay. Next one. The magnification should be stated in the following way when linear or line diagrams are drawn upon observing a specimen. Now, do you remember I told you all, if you are going to do biological science for your A-levels, then there you have to draw the line diagram of the specimen while looking through the microscope. Right, children? Now, when you observe through the microscope, you will see the specimen as if like it's a small circle. But when you draw it on your book, you are going to magnify it a bit and draw. You are not going to draw it in the same small size. You are going to draw a bigger picture of that specimen that you are observing. In that case, you have to consider that magnification as well. Okay, so how to indicate that? So in that case, the total magnification equals magnification of the eyepiece into magnification of the objective lens into the number of times the visual image is magnified when drawn. Now here we can't exactly calculate this one, because while looking at the object or the specimen you are drawing the diagram, you can't exactly say how many times you magnified here, but roughly you can come to a conclusion. Okay, now here the total magnification equals the magnification of the eyepiece and the magnification of the objective lens. Now this is what we learned earlier, only this part. Magnification of the microscope equals magnification of the eyepiece into the magnification of the objective lens. The first two parts given here, right? This is what we learned, only this part we learned earlier as the magnification. But in case you draw a diagram, a line diagram of the specimen, you have to indicate this one as well. So in that case, magnification of the eyepiece into magnification of the objective lens, the number of times the visual image is magnified when drawn. So whether it's 2 times or 3 times, or roughly you can mention that as well. Right, children? Okay, so I hope you understood this part very well now. We'll move on to the next. Right, we have this activity to do in the lab. Observation of plant and animal tissues with a compound light microscope. So far we learned what are the steps to be taken when we use this microscope and all. Now we are going to observe some specimen under the light microscope, compound light microscope. So what are the things we need? We need a compound light microscope. A compound light microscope. And we need glass slides. Slides and cover slips should be clean. Glass slides. These are the cover slips. And we need a leaf of betel. And we need a leaf of royo. Royo leaves a purple color, right? The lower epidermal, the lower side is purple color, the upper side is bit greenish color, right? The leaf of royo. And we need a bulb of onion. And finally, we need a slide made of cheek cells. Now here we are not going to make the slides. Now all these 3 instances we are going to make live specimen of a leaf of betel, a leaf of royo and a bulb of onion. Right? We are going to make the specimens of onion, the lower epidermal of royo and betel, and we are going to observe the skin of onion, and here we are going to use a permanent slide. We are not going to make this slide of the cheek cells. We are going to use a permanent slide here, okay? A slide made of cheek cells. Right? A compound light microscope, glass slides and cover slips, leaf of betel and a leaf of royo, a bulb of onion and a slide made of cheek cells. Right? What to do here? What do we have? Method. Observe the above mentioned types of plant and animal cells or tissues by various magnification powers. There are 3 powers that we can use: low power, mid power and high power. Right? Draw line diagrams and mention the magnification of each of them. We are going to do this activity now. Right, children. Now we are going to observe some plant and animal tissues using the compound light microscope. Now you all have a good knowledge on how to use a compound light microscope, okay? So I am going to prepare some slides of some plant cells and animal cells, right? Okay. So we are going to observe the cells of the onion peel, royal leaf. Now this is the royal leaf. This is the upper side of the royal leaf. It's green in color. This is the lower surface of the royal leaf, which is purple in color, right? So we are going to observe a peel from this lower surface, right? And this is a beetle leaf. Here also we are going to observe a peel of the lower surface of this leaf, okay? And apart from that, we are going to observe human cheek cells as well. There, in order to show you all the human cheek cells, I am going to use a permanent slide, okay? Right. First of all, I'll show you all how to make a specimen using this onion peel. So we need to take a piece of onion like this, and we need to obtain a very thin peel of this one. So I'm going to take a piece like this, and I'm going to break it like this, and there we can obtain a peel very easily, right? So here I have some water in a petri dish. Therefore, I'm going to put this onion peel into this petri dish, so the cells will not dry off. Right. Now here I have a paintbrush, and I'm going to take a slide. This is a glass slide. Now you all know that glass slides and cover slips that we are going to use should be very clean, right? I'm going to take this onion peel to the petri dish now. Right. And you have to spread it like this until there are no wrinkles. And then you need to cover this with a cover slip. Right. Okay. So before you cover it with a cover slip, we need to put a drop of water here. In that way, the cells will not dry off and we can observe them well, right? So how to do this? You have to take a cover slip, and when we make this slide, remember children, we need to make sure that air bubbles are not trapped inside. Therefore, I'm going to hold it like this and hold it at an angle like this, and I'm going to drop it like this. And then we have to give a little tap so that if there are any air bubbles, they will go off, right? And then I'm going to wipe the excess water off this slide. So you all know that we have to always wipe off the slides with a clean piece of cloth or cotton, right? And now we need to mount this. We need to place this on the stage, right? So you all know that before we place this, you all have to make sure that whether the low power is on. Otherwise, if the other high power is on, what happens? Because they are too long, it might damage the, the slide might damage the objective or the lenses, right? Therefore, now the low power is on. Therefore, I'm going to mount it like this. Right. I need to turn on the lights and observe. Right. So my low power is not enough. Using the rough adjustment knob, I'm going to take it a bit down. Right. So I'm going to turn it to the medium power using the right, children. Can you all see the cells? Some elongated cells you all can see. Right, children? Okay. So that's how you can see the cells of an onion peel. Right, children? Now we are going to observe the peel of this lower surface of the royal leaf, right? So I'm going to make the slide in the same way. So I obtain a very small, very thin piece here. Right. I'm going to use another clean slide to mount this one. Right. And I'm going to put a drop of water. So sometimes instead of drop of water, we can put a drop of glycerin as well, okay? Right. In the same way, I'm going to hold it like this and drop. So you all can see there are no water bubbles, it's all clean. And you need to wipe off the excess water. Right, now we need to mount this one. So before we need to mount this one, we need to remove the previous slide. So again, you all know that when you remove this one, what you have to do, children? You need to turn it to low power and then remove the slide. And now I am going to adjust it according to by looking at this one. Observe carefully, children. You all can see a very clear image of the cells. Right, children? Right. I hope you all can see very clearly. Right? I'll put it to the next power as well. Right. You can see a clear, right, children? This time also you can see a kind of elongated cells. Okay, children? Right. So I hope it was clear. Now we are going to make a specimen of this lower epidermal tissue of the betel leaf, right? So we need to take the leaf in the same way. I'll try to tear. Okay, so when I tear it like this, remember we need to obtain a very thin layer. Otherwise we can't. If the cell layers, if there are many cell layers, we can't observe it, okay? So you all can see this thin part. I'm going to take a piece from this very thin part, okay? See? Right. Now I'm going to take another slide and mount this. Right. And then put a drop of water. And with the cover slip, you have to cover it like this. Wipe off the excess water and we are ready to go. This to low power so that you will not harm the lens. And then mount this one. Right. Okay, children, you all can see very clearly all the cells. You all can see those tiny cells, round cells. I will show you all a clear picture of that round cells. Right? Okay. Right, children. Now you all can see bigger picture of that round cell. Those are stomata. You all know that stomata of the plant leaves let oxygen go out and carbon dioxide enter the plants. Okay, children? So you all can see the stomata here very clearly. Right, children? So you observe 3 different types of plant tissues. Onion peel, beetle leaf and the royal leaf, epidermal tissues. Okay? Now we are going to observe the human cheek cells. Now I'm not going to make the specimen with the human cheek cells. Instead, I'm going to make a previously made permanent slide. So in order to make human cheek cells, what we have to do is using a yogurt spoon like this, we have to scrape some cheek cells from the inner jaw and then from the inner side of your mouth, and then you have to place them on a slide, and we have to make the slide in the same way we made this other slides. Okay, so here I'm going to use a permanent slide for that. Right, children. Check whether you all can see the cheek cells, children. We'll put it to the next power. So that you can see it very clearly. Right. Now check whether you all can see, children. It's very clear. The cheek cells are a bit round in shape. Right, children? So they look purple in color. Because now here you can see the cheek cells purple in color because when the slide is made, it is stained, which means colors are added for easy observation. Right, children? So we observed 4 different types of tissues, plant and animal tissues, 3 plant tissues and 1 animal tissue. So I hope this part was clear. Right, children. So you all observed what happened. So at school, you can observe these type of specimen with your teachers, and you can even try to draw the line diagrams of the specimen, right? Here, now this is the microscopic view of the specimen, and this is the line diagram. Here I have the row or lower epidermal tissue under low power of the microscope. So you observed how we obtained this lower epidermal tissue, right? From the lower surface of the leaf, we took the tissue and the row here, lower epidermal tissue under low power of microscope. So 10×4, right, children? So, and this is the linear diagram of the row here, lower epidermal tissue, right? The line diagram of the row here, lower epidermal tissue. You all can see here, this magnification is 10×4. As we explained before, if you draw the diagram, you have to indicate the visual magnification as well, which means 3 times. Now consider this square. So when you consider this square and this square is about 10 times larger than this. That is what mentioned here, right? So visual magnification is about 3 times, okay? So the line diagram of the row here, lower epidermal tissue. So when you see the cells, you observe the cells look like this. Okay, children? Right. Now here I have another tissue we observed. The stained human cheek cells under high power of light microscope. Right, 10 into 40. 10 is the power of the eyepiece, 40 is the power of the objective. Right, under high power. The stained human cheek cells under high power of light microscope. So when you draw the line diagram, look at this square. It's magnified by about 2 times. That's why the visual magnification is about 2 times. Right, so you have to, when you draw the line diagram, you have to mention that as well. So the diagram of the human cheek cells. Right, children? So you can observe other specimen also in the school lab. You can observe the other specimen as well, and you can try to draw the line diagram of those specimen too. Right? Okay, so I hope you understood this part well. We'll move on to the next. We have an assignment to do here. List the differences between the simple microscope and the compound light microscope. We'll try to do this now. You all can try before me, right? Some of these things you have already learned that. Now even I don't even have to teach this part when you consider about earlier. The simple microscope means the hand lens and the compound microscope. Now we already know what are the differences, okay? So you can start this before me, okay? Right. We will put it in a table. It's easy then. Right, children, let's start. Simple microscope. Simple microscope. Compound light microscope. Compound light microscope. Right. Simple microscope and compound light microscope. What is the main difference? We call this simple microscope because only one convex lens is used. There we call it compound microscope because 2 convex lenses are used, right? 1 convex lens is used. That is the first difference. 1 convex lens is used. Here, 2 convex lenses are used. Right? Number 2. What can you tell about the magnification and the resolution? The magnification and the resolution is less in the simple microscope, but here when you compare it with the compound light microscope, compound light microscope's magnification and the resolving power is higher than that of the simple microscope, way more higher, right? Okay. Magnification and the resolution is lower the magnification and the resolution higher. Right, children? Next one. Now, this hand lens. So the simple microscope can be used to magnify the smaller objects that is visible with the naked eyes. Right? Now, when you consider the compound light microscope, you can clearly observe the microorganisms. Are the microorganisms visible with the naked eyes? No. So therefore, compound light microscope can be used to magnify the objects that are not visible with the naked eye. Right? We will write that difference as well. Even though we use a simple microscope, we cannot observe the microorganisms. Can be used to magnify the small objects that can be seen. That can be seen with the naked eye. Here can be used to magnify the small objects that cannot be seen with the naked eye. What is the next one? We can write one last thing. What can you tell about the structure? Here, this is a very simple structure. Just a handle and a frame and a convex lens. What about that one? Compound light microscope. We learned there are many parts. It's a complex structure. Okay? The structure is simple. The structure is complex. So if you can think of other differences, you can include them as well. Right, children? So check whether in the list that you wrote, in the differences that you wrote, these are mentioned or not. Okay? One convex lens is used. So simple microscope and compound light microscope, the differences. One convex lens is used. Here two convex lenses are used. Right? Magnification and the resolution are lower. Here the magnification and the resolution are higher. Right? Can be used to magnify the small objects that can be seen with the naked eye. Right? Here can be used to magnify the small objects that cannot be seen with the naked eye. Even though we can use this simple microscope to magnify objects, we cannot see the objects that we cannot see with the naked eye. Right? So the structure is simple here, the structure is complex. Right, children? I hope you understood the differences between the compound light microscope and the simple microscope. We'll move on to the next. Right, children. This is the last topic of the lesson, the electron microscope. So in this lesson, we are learning about three different types of microscopes. Simple microscope, that is the hand lens. And the compound light microscope also we did. There two convex lenses are used. What is this? The electron microscope. Now in the compound light microscope, a light beam is used to observe the specimen, but here instead of the light beam, an electron beam is used in the electron microscope instead of light rays, right? So when you consider all the objects, everything is made up of atoms. You are going to learn this in the upper grades, right? Every matter is made up of small particles, and these small particles are known as atoms. Now in grade 9, you are going to learn about the atoms. When it comes to an atom contains another 3 different types of particles. Those are known as electrons, protons and neutrons. So here an electron beam, that type of electron beam is used in the electron microscope instead of light rays. Okay children, so here this is extremely powerful than the simple microscope and the compound light microscope. Let's go through these details. The maximum magnification power of an electron microscope is 500,000 times. Do you remember the maximum magnification of the compound light microscope? It's only about 2000 times. This is 500,000 times. 500,000 times. And its maximum resolution is about 0.0005 micrometers. What is the resolution power or resolving power of the healthy human eye? That is only 0.1 millimeters. What about the light microscope? That is about 0.2 micrometers. This is 0.0005 micrometers, right? So this is 200,000 times more than a healthy human eye, right? So the maximum magnification is about 500,000 times. Resolution is about 0.0005 micrometers, right? So this resolution is 200,000 times more than a healthy human eye. This is why this microscope is so powerful, right? So because of this reason, we can observe many tiny objects, right? Extremely small objects using these type of microscopes, okay, electron microscopes, okay. Do you remember we learned that microorganisms except for viruses can be observed using the compound light microscope, because viruses are way too small to be observed by a compound light microscope. So the viruses also can be observed using this electron microscope, okay. So although viruses cannot be observed by a light microscope, they can be observed by an electron microscope. Right, children? Okay. So now we are going to learn the uses of the electron microscope. So most of the time, to observe the activity of pathogenic microorganisms. What is the meaning of pathogenic microorganisms, children? Pathogens means the microorganisms that cause diseases. Now in grade 6 you learn that microorganisms, no, they are not always harmful. There are many uses of the microorganisms as well. So the microorganisms that cause diseases are known as pathogenic microorganisms. Most of the time, viruses, bacteria, fungus, they cause us diseases, even unicellular organisms. Right, children? So pathogenic microorganisms, to observe the activity of pathogenic or disease-causing microorganisms, especially viruses, because viruses cannot be observed under the compound light microscope. Therefore, we have to get the help of the electron microscope. So what are the diseases caused by viruses, children? Dengue. Rabies is caused by rabies virus. Dengue is caused by dengue virus, right? And coronavirus causes COVID-19 likewise. Okay, children? So to observe the activity of pathogenic microorganisms during research, we need the help of the electron microscope. To learn about the internal structure of the cell in detail, now we can observe the cells under compound light microscope. But even inside these small cells, now there are two main types of cells, plant cells and animal cells, Right, children? So inside these cells, there are tiny other structures as well. Right? These tiny other structures are known as organelles. Even though we can observe the cells under light microscope, we cannot observe all the structures under the light microscope, all the other structures. So in order to get a better understanding about the small organelles, small parts present in plant and animal cells, electron microscopes are very important, right? To learn about the internal structure of the cell in detail, right? To use in genetic research. So genetic research, you must have heard of DNA, genes, right? So the children get their specific characteristics through their parents, through the DNA, right? So DNA, that's why like sometimes we say, you look like your mother, you look like your father. That is because those details are indicated in DNA. Right, children? So here to test this genetic research, genes containing the DNA. So to study about these DNA and about the genes, those type of things also, electron microscope is very important because we can't observe genes, DNA through the light microscope. Okay, children, to get information on crime investigation, to find the criminals, so to find out sometimes like to investigate about the crime place, right? The incident, where the incident took place, those type of places, to find out what are the residuals like gunshot residue, those type of things left by the criminals can be investigated using the electron microscope, can be analyzed using these type of electron microscopes. Whatever the body fluid or whatever the other pieces of other parts, let's say metal pieces or those type of anything left by the criminals can be analyzed using the electron microscope, and it's very important in crime investigation because of that. Right, children? Okay, so here look at these two images. Image of a AIDS virus under the electron microscope. Image of a bacteria under the electron microscope. Right, these are only some images. Now we are going to watch a video clip on these type of images given by the electron microscope. Right, children. So I hope you enjoyed that video clip related to the electron microscopic waves. Okay, children. So we have completed the entire lesson. So do you remember what we learned in this lesson? Let's go to the summary and see. Right, the summary. Transparent objects with a convex nature can be used to observe small things in a large scale. Right? So as the first experiment, we used a number of objects with transparent surfaces and with a convex surface. Right? Transparent objects with a convex nature. Convex nature means a certain surface that is protruded or curved outwards. Okay, with a convex nature can be used to observe small things in a large scale, to magnify things. Right? Hand lens, light microscope and electron microscope are three optical instruments used to observe small things in a large scale. Right, so out of these microscopes, the electron microscope is the most powerful one. Right? The magnification of a light microscope is obtained by multiplying the magnification of the eyepiece with the magnification of the objective lens, because the compound light microscope has two convex lenses, right? In order to get the total magnification, you have to multiply the magnification of both the lenses that you used. Okay? The minimum distance that should be present to clearly distinguish between two adjacent points or objects is called resolution or resolving power. The electron microscope has a higher magnification and a higher resolving power than the light microscope. Right, children? So we learned how to handle the compound light microscope, and we observed some specimen also under compound light microscope. So in the next chapter, we are going to do the activities, the exercise. So before you watch that chapter, you all have to answer all the questions by yourselves.
To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.